Hello everyone, I'm Priyanka and today we will discuss about culture media. So this culture media is a special media used in microbiology laboratory to grow different kind of microorganism. Culture media contains nutrients and physical growth parameter necessary for microbial growth. In laboratory, we give artificial suitable environment for the growth of microorganisms such as peptone, meat extract and pH around 7.2 to 7.4. So this is the normal composition of our culture media. Talking about the history, Louis Pasteur was the first one to use culture media. He used urine meat broth. After him, Robert Koch used cut potato. Later on, he used gelatin, but the use of gelatin was not satisfactory as gelatin became liquid at 24 degrees Celsius. Agar-Agar is considered as the gold mark on uh, microbiology. So, the use of agar-agar is suggested by the Frau Heza, wife of one of the investigator in Koch laboratory. As the agar becomes solid at 42 degrees Celsius and melt at 98 degrees Celsius, uh, in addition to that, it don't have any nutritional value. We can use almost 2% of agar for solidification, and it can easily be obtained from seaweeds. Next, we will discuss the classification of culture media. Culture media can be classified as follows based on the agar concentration used, based on ingredient, based on oxygen requirement. So this is example of solid media, liquid media and semi-solid media. Talking about the solid media, liquid media and semi-solid media, in solid media we use 2% concentration of agar powder. Talking about the advantages, bacterial identification is possible. It can be used for the pure culture growth. Talking about the disadvantage, it takes 24 hours to grow. For example, blood agar, chocolate agar. Next we have liquid media. No agar is used in liquid media so that it can remain in liquid state. Talking about the advantages, growth appear within 4 to 6 hours. Talking about the disadvantage, no identification of bacteria mix growth. Example, peptone broth or nutrient broth. Next we have semi-solid media. Now here we will use the concentration of agar powder in around 0.2 to 0.5%. Now this type of media is used to check the motility of bacteria. Next we have aerobic and anaerobic bacteria. Aerobic bacteria used for bacteria which can easily grow in presence of oxygen. Talking about the anaerobic media, anaerobic culture media used for bacteria which can't grow in the presence of oxygen. For example, Robertson cooked meat broth. Meat particle utilize oxygen. Then we have thioglycolate media. 1% of thioglycolate reduce oxygen, and this type of media is mainly used for the Clostridium species. Next we have simple media, complex media, defined media and special media. First let us talk about the simple media. It contains basic simple ingredient, for example nutrient broth and nutrient agar. Okay, so basic composition is peptone, meat extract, NaCl, water and pH around 7.2 to 7.4. Advantage, we use this type of media in regular diagnostic in laboratory and for the maintenance of stock. If we add 2% of agar in nutrient broth, it will become nutrient agar. Okay. Next we have complex media. When we add some extra nutrient substances to simple media, it become complex media such as blood, egg, serum, etc. The example of complex media are blood agar, Lg media, Lufer serum, slow media. Next we have defined media. This type of media contain pure cultures, pure chemical substances, exact composition, exact concentration of chemical substances used. Um, for example, peptone water. Peptone is 1% and NaCl is 0.5%. We use this type of media for research purposes. Other than that, you will heard about the semi-defined media. Now, in this type of media, the exact chemical composition is not known. In this type of media, concentration of chemical is approximate but not exact. Next, we have a special media. This media contains special substances for the growth of bacteria. So, following are the special media, list of the special media we use in laboratory: enrichment media, enriched media, selective media, indicator media, differential media, transport media. First of all, let us talk about the enrichment media. This is liquid in nature, called as a liquid selective media. This media favor the growth of the particular bacteria either by adding enrichment substance or by adding the inhibitory substance which slow down the growth of unwanted bacteria. For example, tetrathionate broth, which contain inhibitory substance bile salt, which inhibit growth of coliform and gram negative bacteria but not salmonella. Then we have selenate F broth. Now in this selenate inhibits the positive bacteria gram positive bacteria and gram negative bacteria, except the salmonella and shigella. Next we have alkaline peptone water, where the pH around 8.6, which will enhance the growth of Vibrio cholera. Next we have enriched media. Now the simple basal media made enriched by addition of one or more nutritional substances such as blood, egg, glucose, etc., which support the growth of wanted pathogen only. So blood agar support the growth of Staphylococcus species, chocolate agar, hemophilia influenza, and Lofer serum slough diphtheria. Next we have selective media. This media contains substance that enhance the growth of selective bacteria and slow down growth of other bacteria. Thiosulfate, citrate, bile salt, sucrose agar. Now in this type of media, thiosulfate inhibit enterococcus, then bile salt inhibit gram positive. Now the alkaline pH in this will enhance the growth of Vibrio. Next we have Lg media, malignant green dye, which avoid the growth of the contaminant, thus enhance the growth of Mycobacterium tuberculosis. Next we have indicator media. This media contain indicator that change color when bacteria grow in them. Wilson barrier media. In this, sulfite get converted in sulfite get converted into sulfide, like black metallic growth is seen on the plate, like the growth is shown by the Salmonella typhi, black metallic. Next we have blood agar, give indication based on hemolysis. Okay, like depending upon the alpha, beta, gamma hemolysis, you will get pink color pigment. Next we have defined media. It is a media which help in next we have differential media. uh, it is a media in which the distinguish can distinguish between bacteria, like McConkey agar distinguish based on the lactose fermentation, lactose fermenting bacteria, pink colony, and non lactose fermenter colorless media. Next we have transport media. It is used to transport the organism to the laboratory from collecting area. Media contain buffer and is non-nutritional. It preserves the particular type of bacteria, prevent the growth of unwanted bacteria. Now we have stored media and carry balloon media. So this carry balloon media for the Salmonella, Shigella, and Vibrio cholera, and stored media for the transportation of gonococci. So that's all we have in culture media. Thank you and have a nice day.